Howdy, everyone. Welcome to the Plant Party, Planting a Seed, hosted by Texas Parks and Wildlife Department, Texas A&M AgriLife Extension Service, and USDA Natural Resources Conservation Service. Plant Party is a quarterly webinar series intended for advanced plant training and fun education. I'm Charles Kniper, State Resource Conservationist with USDA NRCS. My co-hosts are Tim Sigmund, the Private Lands Program Leader with Texas Parks and Wildlife Department, and Megan Clayton, Professor and Extension Range Specialist with Texas A&M AgriLife Extension Service. We have 23 wonderful door prizes we will be giving away. Our door prize donors today are committed to supporting you in your quest to learn more about plants. I'd like to thank Bamert Seed, Corteva, InView, formerly Bayer Crop Science, Renewable Resources Extension Act, Texas Brigades, and USDA Natural Resources Conservation Service for donating these neat prizes.
Thanks for that intro, Charles. So let's get some housekeeping out of the way. We'll be moving quickly through 5 presentations today. Our first presenter today is Dawn Stover. Dawn joined the USDA Natural Resources Conservation Service in November 2021 as a study leader agronomist at the East Texas Plant Materials Center in Nacogdoches, Texas. She is currently working on germplasm adaptation trials, conservation plant releases, and seeding rate studies to benefit landowners and land managers in the western Gulf Coastal Plain. Prior to her current service, Dawn spent more than 2 decades as a research associate with the SFA Gardens at Stephen F. Austin State University, Action Jacks, where she developed an extensive program to collect, propagate, display, and distribute plants native to the piney woods of East Texas. She has extensive greenhouse and nursery experience and a focus on growing plants that are beneficial to pollinators. She enjoys sharing her knowledge and experiences with the public through talks, tours, and in-person demonstrations. Dawn will be presenting on seed terminology today. And thanks for kicking off our plant party, Dawn. Take it away.
Good morning, everyone. Thank you for having me. Well, today I'm going to talk to you a little bit about seed terminology. This is probably not one of the most romantic things we can talk about in the plant world, but it's important, and it's important to know what we're working with and what we're using. So here we go into the world of seed terminology. I'm going to start you all off with a quote from Henry David Thoreau. Though I do not believe that a plant will spring up where no seed has been, I have great faith in a seed. Convince me that you have a seed there, and I'm prepared to expect wonders. I absolutely enjoy one of the most things that I love about my job the most is watching seeds germinate out in the field and in the greenhouse. It's just a really wonderful testament to life. Really didn't want this presentation to turn into a glossary of terms, but I think that's the direction that it needed to go. So hopefully by the end of this 15 minutes, you'll leave with a better understanding of seed terminology and maybe even learn a word or 2 in Latin. Yeah, we do have to go there. We have to talk a little bit about botanical nomenclature. This is the scientific naming of plants, and it is governed by the International Code of Nomenclature for algae, fungi, and plants. We use a binomial system consisting of genus and species that was formalized by Linnaeus in 1753 in his Species Plantarum. And we don't throw Latin names at folks to sound pompous or arrogant or to make us look smart. This system of nomenclature or classification is consistent worldwide and adheres to the ICN code. Scientific names are unique to each plant, and that's something that we need to take forward with this, is the fact that this is a unique name for each different plant. Let's talk about the case against common names. I'm not opposed to common names per se, but there is often a great deal of discrepancy with them. Sometimes the same plant can have many different names. They'll have more than a few different common names, and it kind of depends on the region sometimes. Sometimes it even just variations on the spelling for the same particular name. There is no rule book, there's no ICN to provide consistency here, and names can differ from one county or parish to the next. So for instance, if we take Erythrina herbaceae, it could be coral bean, or Cherokee bean, or red cardinal, cardinal spear, and if you're in southern Louisiana, it might be known as mamu. So there's a lot of confusion here because there's no consistency with a particular common name. Sometimes the same name can refer to many different types of plants. So the name lily is used for no fewer than 200 different common names. In our slide here, we have the pinewoods lily, the crinum lily, the voodoo lily, the daylily, tiger lily, rain lily, pineapple lily, spider lily, gloriosa lily, lint lily, and the Philippine lily. And only 2 of these particular plants in this slide are actually in the lily family, so only 2 are true lilies. And the common names have language restrictions. As we've seen, they aren't consistent because they're not standardized. They're different in different languages. For instance, purple coneflower, of course, in English, is known in Germany as Lila Sonnenhut. So there's a lot of, it's lost in translation, basically. But in any country, they are called Echinacea purpurea. And just like in slang terms that can change over time, common plant names can change too. I grew up calling a particular plant the mother-in-law's tongue, but in this day and age of the revival of houseplants, they're calling that the snake plant. So here we are back again with binomial nomenclature. We're going to refer basically to the genus and species. I know we all remember in biology class in the 9th grade, remember learning that King Philip came over from Germany singing, which is the kingdom, phylum, class, order, family, genus, and species. And these are the levels of botanical or biological classification from the most broad, from kingdom, to the most specific with species. When we're referring to plants this way, we're only going to kind of talk about the genus and the species. And you can think of the genus as a group of related plants. They're like all of your cousins. They're ranking between family and species, and they're grouped based on shared attributes. They have more in common with each other than plants in the family level. The name is always capitalized and it's italicized or underlined if you're writing in script. So we're going to take our example with Echinacea here. And then we narrow it down. Species is exactly what it sounds like. It's specific. It's the description of one specific plant within a genus. It is the most basic unit in the system of classification. And species have certain characteristics that differentiate it from other members in the same genus. What's important to remember is that 2 members within the species must be capable of producing fertile offspring. And the name is always written in lowercase, and it's either italicized or underlined. So our example here is the little plant to the right, which is my favorite flower. This is Echinacea sanguinea. So I know that commonly I call this the sanguine coneflower, but it actually goes by several different common names. So knowing those unique identifiers is beneficial to knowing the plants specific to your ecoregion. We work a lot with native plants, and we want plants that are native to our ecoregions and the correct ecosystems, especially when we're working in conservation activities. Our goals are to choose the right plant for the right place. So commonly, I see Echinacea purpurea, or the purple coneflower, listed as a native plant for Texas. And if you look at the range map here provided by USDA plants, Echinacea purpurea only actually occurs in less than a handful of counties in Texas. So it's kind of a misnomer to say that it is a native plant to Texas. It is native to the US and native to a lot of the Southeast, but not necessarily an appropriate Texas native. We can take that a little bit further with the Biota of North America program. They have there's this same kind of mapping system, but on their website, you can pull up an entire genus and see the different species in a different range. So I'm not great at botanical identification. I'm really not good at keying plants out, but generally I can get it to the genera. And if I pull this range map out, and I know that I'm in Nacogdoches County and I can see which echinacea might occur in my county, I know that it's probably going to be Echinacea sanguinea. It's not foolproof. Sometimes the species aren't always listed on the maps, but it's still a very useful tool to kind of get you to where you're going. I'm often asked the difference between a cultivar and a variety. A variety is naturally occurring variation within a species. It shares most characteristics of the species, but with a notable distinction. The population is uniform, and the seeds from that population will retain the characteristics of that parent. The name is written in lowercase and italicized, and it follows a VAR period after the genus species. So you can see 3 different botanical varieties of Gaillardia astabilis here. Variety astabilis is what we know as the species. Variety flavivirens in the lower left corner has yellow discs and yellow ray petals. And then the variety Winkleri is actually pretty variable, but this one here shows purple discs and purple rays, but they're all within that species, Gaillardia astabilis. These are just botanical varieties, and they each do come true from seed for that particular color trait. Moving on to cultivars. So cultivars is a cultivated variety. It is selected. Man has had to have his hand in this particular selection process. They are either selected or bred by humans. They can include mutations, sports, and hybrids. You can think of a mutation as basically if you have a leaf that comes out variegated, or a sport might be a white flowering branch on a red flowering azalea. And of course, hybrids are intentional crosses between two different plants. They're most often clonally propagated. That means they have to have the very exact same genetics to keep those particular traits that they were selected for. The cultivar name is always capitalized. It is a proper name. It's never italicized, and it's surrounded with single quotes. And these are governed by the International Code of Nomenclature for Cultivated Plants. So you see I'm going back to governing bodies that regulate these things. There's a consistency with the botanical nomenclature. When we move from horticulture into conservation planning, we need to start at the very beginning with an accession. This is not necessarily a term that you need to know going forward, but it's where we start. It's the most basic building block into our conservation releases here. So there's plant material that's collected and assigned a unique number. It can be a plant, a seed, or a vegetative part like a cutting. The identity is maintained throughout evaluation, increase, and storage. So it's not necessarily, you don't necessarily need to know the accession, but you need to know that this is where it starts. So this particular collection of Virginia wild rice seed, Elamis virginicus, will receive in our system a 9 million number. And that's the accession number that will go forward all the way through to increase in evaluation and selection. Okay, so my particular service region is Major Land Resource Area 133B. It's also known as the Western Coastal Plain. This is the ecoregion that I focus on when we are working on our conservation releases. I want plant materials that can adapt to our soils, adapt to our climates, that can tolerate heat and humidity. I don't necessarily want plants in conservation purposes and restoration work that come from Kansas or Alaska even, because they're not going to persist and do well in our ecosystem here. So this will come up in a little bit, but this is where our accessions focus on when we work on conservation plant releases. So it really basically starts out in a brown sack when we're collecting plants in our resource region for conservation releases. They'll start it out in a bag like this. They'll receive a 9 million number. We'll keep track of where they came from, the GPS coordinates, the soil types, and things like that. And all of that eventually ends up into our release process, like the Coastal Plains Germplasm release brochure that you see on the right there. In conservation work, we do work with cultivars. Nacogdoches Eastern Gamagrass is a cultivar level release. It is a single accession from a single collection that was evaluated for specific performance. We don't manipulate the genetics here. We are basically selecting for a known level of performance. Currently, the plant materials program has moved away from cultivar levels in order to include more diversity in the genetics of our releases. Let's talk briefly about source identified seed. We don't typically work with that, but we'll, in certain instances, we at the East Texas Plant Materials Center partnered with the U.S. Forest Service to grow pine hill bluestem for conservation planting. This is actually a botanical variety of little bluestem grass that is a little bit more shade tolerant and works as a fine fuel source in longleaf and shortleaf pine ecosystems. And that shade tolerance is important as the trees grow. This particular variety is not available commercially. At least it wasn't several years ago when we were working with the Forest Service. So that's just the application that we would use that source identified seed. And finally, let's talk about germplasm. Germplasm is governed by AOSCA, which is the Association of Official Seed Certifying Agencies. It's tested and certified. It is regionally adapted. It's selected for performance and production, and the name is always capitalized and always includes germplasm when it's written. So this Coastal Plains germplasm little bluestem is actually a combination of 77 different accessions from my resource region 133B. And it regularly outperforms other commercially available selections that were developed outside of the southeastern U.S. So it works very, very well in eastern Texas and western Louisiana up into the southeastern parts of Oklahoma and southwestern Arkansas. Talk briefly about VNS, and I'm going to quote from the American Agriculturist. VNS designated seed is notorious for being anywhere from the least costly to the best performer to having the worst germination and a weedy mess. Basically, when you get VNS, you don't know what you're going to get. And I know I'm going really fast, but I need to get to Pure Life Seed because that's probably the most important takeaway that I think y'all should have today. So Pure Life Seed, we strongly advise purchasing seed from a company that performs regular seed lot testing and can provide a percentage of Pure Life seed for the product you select. So you can see in that red circle, this particular company has provided you with a percentage of Pure Life seed, 76.95%. To find the percentage of a viable seed in a given seed lot, we take the percent purity times the percent germination, and that's where we get the percent PLS. So in order to find out how much of the bulk rate we need, we take the PLS pounds required. So if I need 20 pounds of PLS seed, I divide that by the PLS percentage of the seed lot that the seed company provided for me. And so I know that with that little bluestem in the previous slide, if I have 20 pounds of PLS seed, I divide it by that PLS percentage, and I need them to ship me 25.99 pounds of bulk seed. So that's kind of that in a nutshell. I know that was really, really, really quick, but basically, if you are working with a reputable company, they're going to charge you for the amount of Pure Life seed you're buying instead of the bulk seed. Talk to your field office for more information, and don't hesitate to reach out to your Plant Material Center's staff too to help you kind of work through all of that and go through that in a little bit more of a leisurely pace. Thank y'all.
Awesome. Thanks so much, Dawn, for that presentation. So to continue this theme of seeds and seed development, we invite Shelley Maher with the USDA NRCS Plant Materials Center in Kingsville to give us a look at the Plant Materials Center's function and overall what they do. Shelley lived under the East Texas pines and red oaks until she relocated to South Texas to attend college at Texas A&M University, Kingsville. There, she received a bachelor's degree in agricultural science, horticulture, and a master's of biology after completing a thesis project on the complex mating system of a rare endemic lily. Shelley has worked as a research scientist with the USDA Natural Resources Conservation Service Kiki de la Garza Plant Materials Center in Kingsville since starting as a student worker in 2002. She was selected as a manager of the PMC in 2021. The research at the Plant Materials Center leads to commercial seed releases and technology for restoration and conservation across South Texas. Shelley, I'll turn it over to you to give your presentation, a look at the Plant Materials Centers.
All right. Good morning, everybody. I'm going to tell you a little bit about the Plant Material Program and the conservation plant releases we make. There's currently 25 plant material centers across the United States. Again, we're more located based on resource areas than we are state lines. Texas is particularly lucky, thanks to our diversity. We have 3 PMCs in the state. And our mission is to deliver needed plants and plant technology throughout the United States to address resource concerns. Again, that plants for resource concerns is the key there. So these releases, we evaluate and release the plants to again, address something. We're not just releasing that individual species because we think it's neat. And the program itself currently has over 600 releases, again, looking to solve those conservation issues. How do we do this with about 3 staff per PMC and maybe a couple student workers? We like partnerships, and we will partner with lots of like-minded groups working on different species or projects or technologies, graduate students, universities, research areas. And this assortment of logos here just gives you a variety of some of the ones that just us 3 Texas PMCs have worked with in the recent years. One of those key partnerships has been the Texas Native Seeds, and you'll hear from them a little bit later. Starting as South Texas Natives back in 2001, we partnered immediately with that. And their goal, again, you can see here, economically viable sources of plants and seeds, restoration, sounds kind of a lot like another mission statement you just heard. So this was an instant and excellent partnership and has led to a lot of releases since then. And it's been a great partnership. So again, when we're looking at those species, we're not just going, oh, I think this one's pretty, or I think this one would be interesting. We're looking for something to solve a resource concern. So for example, TxDOT came to us in Texas Native Seed years ago and said, we get complaints about using invasive exotics on highways. What other species could we use? What natives could we use? So what we look at is the characteristics of the plants that are currently existing there. And then we look for native species that would have similar characteristics or that can compete in that environment. And if you ever heard a talk by the former manager, John Riley, you would always hear him say, can you harvest it? Can you grow it? Can you maintain it? These species that we deal with are going to have to be commercially viable. If, again, if it can't be grown with height and weed competition, if we can't harvest it with commercially available equipment, and again, we've got some interesting stuff we can use, but it's got to be something that can be harvested. And then can we process that? Seed fill in particular is an issue in South Texas. Can we get this clean enough to be useful and to plant? Can we get those seed numbers up high enough? And if we, get a couple of collections and look at it out in the field, and the answer to these questions is no, then we capture that information in a plant guide. It goes up on the plant database. The research isn't lost, but we don't then try to get this out commercially and then it fail. We just kind of stop at that point and collect the data. But if that species does show promise and it is going to be good for a resource concern, then we do a study plan, we do a literature review to see what other research is out there, and then we make a call for collections. Again, Texas Native Seeds has brought in the most collections we've had, but we get some from the NRCS field offices, from wildlife preserves and refuges. Fish and Wildlife has been an excellent partner, and some individual landowners send us collections. Depending on the species we're looking at and how common it is in our area, we try to get 10 to 100 collections of that. We want to get good representation. We want to take a good look at this thing. And sometimes the more challenging species, we need more collections to look through and sift through. When we start collecting, you can get into some groups and some arguments about what's native, what's genetically local, and that sort of thing. Again, we're kind of looking at our ecosystems, our ecoregions, our ecotypes. We want to look and consider the breeding system. Is it a short-lived annual? Is it a long-lived perennial? What's the seed dispersal on like this? How far does that seed go? The successional stage of that species can make a difference. Topography in places that have it, we lack that here, but there are places in your mountainous regions where topography can make a collection of a seed zone significant. And now we're starting to incorporate more genetic testing in this as well. And in that genetics, we want to look at the genetic adaptability. We don't want something that's, just only useful in a tiny little space. We want it adapted across the range where it's going to be used. And is this a species that is prone to inbreeding or outbreeding depression? We can get into some weirdness with the wild rice pretty quickly. So we try to take all this stuff into consideration when we start making those collections and put some good science behind it. So after a collection comes in, we clean it up. We give it those accession numbers Don was talking about. Stick it in the vault. Our storage is not the long-term storage like you hear the millennial seed vault, that kind of thing. It's kind of refrigerator. We're looking at it's 5 to 20 year storage, not, centuries. Then we take those seeds, once we get enough collections, we put them in the greenhouse. We take some germ counts, see how they look in the greenhouse. Anything that's got enough seedlings, we take then out to the field and we put it in what's called a common garden study. We take these accessions, again, from collected all over our ecoregion, put them out there side by side and see how they perform. And these are the characteristics we're looking at: survival, vigor, seed production, seed shatter. Does the seed even stay on the plant long enough for us to harvest it, or does it fall out quickly? Is it disease resistant? Can it handle a drought? In South Texas, that's an important thing. And then we go out there with our little lunch sacks and Sharpies, and we collect the seeds off of those different accessions. We bring those in, we weigh the quantity of harvest, we run germination tests, lining those little seeds up on the papers there. I enjoy that process, not everybody does. And then we evaluate this, both the plants in the field and then the seed germination. We evaluate that for several years. 3 is kind of the minimum, longer if it starts to be a challenge, if there's a particular characteristic that's an issue. And then when we can, we try to do offsite evaluations as well. Send them to other plant material centers, send them to some of the offsites by Texas Native Seeds or other research folks, and see how it does in places besides just Kingsville. And now we're starting to incorporate genetic studies. Now that technology is becoming more readily available and more affordable, we'll be able to see a bit more about the genetics we're actually using there. And then based on how they do, a top 1 accession or multiple accessions will be chosen for release. In that release process, once we've chosen it, we do a botanical verification to make sure it really is what we think it is. Clear the name, we do an environmental evaluation. Again, most of what we're dealing with is a native species, so it's not going to have a bad impact on the environment. We do a final study report, we write up the release notice, do a plant guide. Again, those plant guides are going to go up on our website and then up on the plants database. We do a release brochure that kind of sums up the information that's kind of more useful for a landowner type use there. And then a native plant journal article to do more of a scientific perspective. And what we don't want to forget in this process is the seed increase. We want to make sure and try to bump that quantity up so that once we say, hey, we've got this new release, then there's something available for the growers. And that's something we're working on. And in that process, we need to make sure there's a grower interest. If it's not a species the growers think they can make, then it's going to be a difficult sell to them. So Texas Native Seed has been an excellent partner in that and keeping in touch with those guys and seeing what those interests are. And we do a seed transfer agreement with them if there's interest. So there's a couple of different release strategies, as you heard Dawn talking about earlier. Cultivars are a long process and typically not what we're looking for. We're not looking for intense breeding most of the time, unless there's a particular species and problem we need to solve. Tested or then selected, and most of ours are selected. Again, that source identified, we don't really do so much anymore. We're mainly looking at that tested or selection section, and we use the selecteds. Our particular PMC takes a couple of approaches to this. We may have a multiple accession approach in that composite, and then we're trying to get accessions from different geographic areas, different soil types, and again, the top performers off of those. And it's kind of that shotgun approach. If we have some good stuff from across the region, chances are something in that is going to work well at your site. These accessions are mixed before the seed is sent to the growers. And this method was particularly useful, again, before the genetic studies were more available, because we were kind of basing our adaptability on what we were seeing in the field, not so much making sure we're capturing that genetic diversity. But now we'll be able to look at that a little closer in the coming years. Another perspective is a single accession release. And we do that if there's one accession in that we look at that really has a different type of characteristic or a particularly useful characteristic. So this is an example of the composite. We were looking at silver blue stem. We had 40 collections. And as we started looking at those, we found out we were dealing with 3 different species. And 14 were silver blue stem. They were very similar. All those characteristics, again, out in the field, germination, very similar. That would have been good for a composite. We had one accession that had very dense foliage compared to all the rest of them. I mean, you could see it from a distance. And that would have been better for either a single accession or proceed to cultivar if you were looking for forage production. So what we did was we did a 5 accession composite, and we pulled that one, and it turned out to be a long spike silver blue stem that had that dense foliage. And then we pulled 4 others that kind of gave us a geographic range. And you can see the original collection locations there in yellow. This gives us a bit of range across our ecoregion. And again, a couple of different soil types, hoping to get that good coverage and good composite there. For the single accessions, windmill grasses. We looked at 43 windmill grasses from across Texas. We did evaluations, Beeville did evaluations. We took these offsite and looked at them, again, looking for all those characteristics, and kind of narrowed it down to 2 hoodeds and 2 short spikes that were the top performers, and did an advanced evaluation. And really looking at it, this ended up being a good for a single release. There was one particular hooded that had stolons. It spread by stolons, and that's not typical of that bunch grass. So for the resource concern we were looking at it for, which again was roadside planting, erosion control, that stoloniferous characteristic was actually quite useful and would be helpful for vegetative spread. So again, to address that particular resource concern we're looking at, this single accession was better than doing a composite of a more bunch grass type. And then we pulled one of the short spike windmill grasses. Again, this seed production on it was kind of an issue. And then we got to where we started looking more at it. And this one had 60% active germination. And then it had about 40% dormant seed. Dormant seed is challenging in the greenhouse. It's challenging in a seed increase field. But once these things are put out in nature, that dormant seed's kind of an insurance policy for our random rainfall events. So it's not always a bad thing to have some dormant seed there, as long as something's sprouting. Now here's the one we had to get really creative. Plains bristlegrass. We looked at bristlegrass forever. We looked at over 100 collections. The germination percentages would range from 0 to 2, 0 to 2 over and over again. They looked great in the field, but just the germination was terrible. We looked at it. Again, Beeville looked at it. Texas Native Seed looked at it. And out of those 100, we narrowed it down to 5 that had some seed potential. We did offsite evaluations all over the place, again, trying to look at this and see, what problem, where maybe this might grow better. But seed fill was the issue. Again, it just had a ton of empty seeds. We worked a bit on some technology to clean it up a bit. We tried all sorts of dormancy breaking techniques. And finally, with these 5, we narrowed down to 4. And you can see here, the viability isn't great for some of them. And the active germination is terrible for some of them. And that 820 in particular only has half viability, only 3% active germination, but it had almost twice the production of seed harvest. So you don't really want to lose the seed harvest characteristic, and it looked fantastic in the field. But then you get to the active germination of 3%, which is never going to work. So again, we got creative. We did 4 separate seed releases. So each of these can be sold on their own, or they can be blended as a commercial blend name. Again, this is not the seed release name, but it's a commercial blend. And then when you blend those together, you have a couple that have high active germination. You have some that have some dormancy. You have a couple that have much higher seed production. So each of them bringing in good characteristics, but none of them containing all those characteristics on their own. And then these are grown separately by the seed dealers. Then they're combined before they're sold, but they can be sold individually as well. And the guys at Texas Native Seed have told me that all of those 4, each of them will perform better in a different area. And there's really not one of those 4 they would recommend dropping. Each of them has their place in the environment. So it sometimes is a challenge on these things. So I hope this has piqued your interest in the release process and our conservation plant releases. Again, the plant guides can be found on the plants database. Our conservation plant releases and their brochures can be found on the plant material program database. And if you've got any questions, have any interest, want to come out for a tour or talk to us about a training session, give us a call.
Thank you, Shelley. That was excellent. I appreciate that presentation. Next up, we have Hagen Meyer, a research technician for the Central Texas Native Seeds Project in Stephenville, to tell us about hand collecting native seed. Hagen has been a part of the Central Texas program for 1.5 years. Previously, he's worked for 2 years for the West Texas Native Seed Program in Alpine, where he received his bachelor's in range management from Sul Ross State University. Welcome to Plant Party, Hagen. I'll turn it over to you.
Thank you, Dr. Clayton. Like she said, my name is Hagen Meyer, and I'm a technician with Texas Native Seeds, and I'm going to be talking to you all about hand collecting native seeds. Texas Native Seeds helps to develop native seeds for the commercial seed market, and in order to do that, we have to go and collect seed from those native sources around the state. We use a few resources to plan out our collection trips. One of those is the Advanced Hydrologic Prediction Service's Observed Precipitation Map. It's a map that will show the accumulated precipitation for a certain time range. And this really helps us select areas in which we can normally expect to find good plant growth with mature seed. So this really helps us choose our areas to go and explore and make collections. We also use historic plant databases. You can find many of these in a real general sense in your plant identification books, but you've got your online resources, the USDA plant databases, historical distribution maps, and and BONAP's historical distribution maps. You can also look at your soil surveys. The UC Davis developed the Soil Web map layer for Google Earth that you can download and input into Google Earth to explore the different soil series around the state. And with that, you can click on each soil series to pull up the NRCS's soil surveys, which will give you a list of common plants that are typically associated with those soil series. So this is another way just to narrow down those areas in the state that you can expect to find certain species that you might be interested in. Also, we use road atlases mostly to find good county roads. We like the real windy ones with changes in topography that go across creek crossings. Those ones you tend to find very diverse plant populations. So we do make a good majority of our collections off of county roadsides. They're normally not seeded with commercial seeds. It's all native stands, or commonly there's a lot of invasives on them as well. So you want to try to avoid the invasives. But your creek and river crossings, you can find your riparian species along those. Cemeteries and historical marker pull-offs are also good sites that we've had good luck finding native seed sources. Your railroad right-of-ways have plenty of space, and they're usually left unmowed, so those are good sites as well to make native seed collections. And then, of course, your private lands. Do be careful with invasive monocultures. They tend to get into your seed collection, so take care when you're collecting your native seeds to avoid contamination with invasives. Also, take note of on TxDOT right-of-ways, it's your highways and interstates and farm-to-market roads. They reseed their roadsides pretty often. On these next two slides, you want to take note of some of the native species that they reseed to avoid making collections of. And each of these links, they'll pull up a more detailed list of your seed, of the TxDOT seed mixes, and they'll have them split up based on districts or your different regions in the state. But this is some of the equipment that we use when we go out. We bring along a GPS to mark the locations of those collections in case we need to revisit them later on. Your field guides to help you identify, and road atlases to get you where you're going. Then your envelopes, clippers, paper bags, gloves, and pins to help you to label and make your collections. Then bring some protective footwear. There's no telling what you'll come across. I've ran across a number of snakes in the grass and brushed my legs through plenty of thorns and cactus. So that'll be a good thing to bring along with you. But once you find your target species you want to collect, you want to make sure that the seed is ripe. And a good indicator of that is the top of the seed heads will begin to shatter, and then the bottom will typically hold on to your ripe seed or seed that is very close to becoming fully mature. So collect these seed heads when you see them. And you may come across some species that seed will mature at very indeterminate rates. When you come across these, if you have the time and ability to revisit these sites, it'll be a good idea to come back and make multiple collections of mature seed off of them. And if you can't quite tell whether the seed is matured just by looking at the seed head, take a pinch of some of those seeds off of the seed heads and rub them in your palm to remove the caryopsis. You observe the caryopsis to determine the ripeness of the seed. In the milk stage and soft dough stage, it'll have a opaque white greenish color to it, and it'll smash easily between your fingers. That's the stuff you don't want to harvest. It needs a little more time. The hard dough stage, this is the stuff you want to harvest. It will have a little give when you squish it between your fingers, but it maintains its shape and form. And then once the caryopsis or seeds reach full maturity, like the variety of these species that are shown on the screen, that's all mature grass seed with hardened caryopses that are been dried out and are good to harvest and will produce pretty good seed later on down the line. With your forbs, you just take a look at the seed heads. Your immature seeds will have a greenish tinge to it, and will hold on to those flower parts. The flowers will still be attached to those seeds. Your ripe seed will be indicated by dry or desiccating petals from the flowers, or no petals at all, and dry stems. And of course, if the seed heads are beginning to shatter or drop their ripe seed, that's the stuff you want to collect. And you can collect them through different methods. You can hand strip them or shake them into the bag directly, or you can clip the seed heads. Probably the cleanest ways to collect are hand stripping and shaking them into the bags. You'll come out with a mature seed with very little chaff, trash in there. Clipping the seed heads, it'll save your fingers, but there'll be a lot of cleaning to be done later on down the line. So when we're collecting, we tend to follow the take half, leave half. We want that population to remain there, so allow it to reseed itself, because you never know, we may need to revisit sometime in the future. And you want to collect a representative sample of the population. Don't just go for the biggest plants. Get as many plants as you can. Try to collect from at least 30 plants and from a variety of sizes in that population, to ensure you are getting a representative sample of that genetic diversity within that population. And you can consider the same species to be the same population if it's within about a half-mile radius. Anything beyond that, you kind of get into the weeds whether or not it's the same population or not. So put a little distance between the two if you need to make collections of the same species and they're on the same road down the road. Put at least a mile or so distance between those two collections. And if you are wanting to send in your seed to the Plant Materials Center, they'll have you fill out this NRCS ECS 580 collection envelope, just to provide information on the collection you made for research later on. Store your collections in porous material that'll allow airflow into the bag and reach the seed, and allow moisture to escape to prevent molding. But if you do collect an especially moist collection, you want to come back a few times and turn it over, just to make sure that it's drying correctly. It won't develop any mold. And store it in a cooler, dry location. Avoid areas that that'll exceed 90 degrees if that's kind of that threshold where seed quality will drop significantly, almost completely. So make sure your collections are stored in a cool and dry location. So these are the plant material centers that we send our collections into. If you're interested in if you're interested in making collections and sending them into the plant material centers, get in touch with the folks at each one, or whichever one you're closest to, and get some guidance from them. I've got their mailing addresses on there in case you are interested in that. So thank you very much.
So we'll move into the presentation on the Texas Native Seeds Program by Dr. Tony Falk. The Dan Duncan, Director of Texas Native Seeds, a program of the Caesar Kleberg Wildlife Research Institute at Texas A&M Kingsville. Dr. Falk has been with the Texas Native Seeds Program for 12 years, working his way up from the Evaluations and Collections Coordinator to his current position. It's all yours, Dr. Falk.
Thank you very much. Thanks everybody for listening. I am the director for the Texas Native Seeds Program, located here or based here out of Kingsville, a program of the Caesar Kleberg Wildlife Research Institute. So our Texas Native Seeds model is a huge collaborative effort. As you've kind of heard from both Dawn and Shelley, we try and work with as many people as we possibly can to be able to do everything that needs to be done. We work with the 3 plant material centers in the state, the 2 that you've already heard from, and the third is the Knox City Plant Material Center, based up in Knox City. Their service area is kind of North Texas up through the Panhandle. We also collaborate with a number of non-governmental organizations, or kind of more traditionally known as NGOs. Those range in everything from the Nature Conservancy, Wildlife Habitat Federation. We also work with private entities. Lavaca Navidad River Authority is one that we do a lot of work with down in the Coastal Prairies area. We work with state agencies, AgriLife Research, AgriLife Extension, Tarleton State, Caesar Kleberg here at Texas A&M Kingsville, Sul-Ross, and then private ranches as well. Railway Ranch, Sambrock, Dallas area, Boggy Slough out in East Texas. And so this huge collaborative effort allows us as an organization, as a partnership with all these different entities, to be able to do the work that needs to be done in evaluations, collections, seed increase, and the whole gamut is needed. If any one group or organization was going to try and do all of this by themselves, the infrastructure that would be needed would just really be unfathomable at that point. So we need to all kind of work together to be able to do these offsite evaluations at different locations, have access to the native plant material that's needed, and really just be able to get the work done. And as some others have shown or discussed today, our mission is to develop and promote native plants for restoration and reclamation of private habitats on public and private lands. This is a very direct mission statement. Our whole point of what we do day in and day out is to develop plant material for restoration and then promote those plants and through use. That drives what we do every day. That's what we all focus on. It does give us the ability to go out and do research into how to do restoration. That doesn't do us any good to come up with all these plant releases if we can't help the public or land managers or whoever wants to use that plant material to figure out how to do it. Everybody would lose interest in doing it, and there'd be no point in having a whole program dedicated to developing releases. So how do we go about doing that? Well, we've broken the state into 6 project regions. Loosely, they're set up on ecoregions as shown here in the map. Each of the different 6 project regions has a regional director in that region. The stars dictate kind of where those people are at. The one star that has kind of been broken off or left off this map is our Coastal Prairies region. That assistant director is based there in Edna. Each of these regions has a team of folks to go out and do the work that we need to do. So we follow the same seed source development model that Shelley had just kind of talked about through her presentation. We identify species of interest, make collections, evaluate those, produce seed, and then work with the commercial growers. The way we identify the species of interest is we form technical committees within each region that we work in. These technical committees are made up of land managers, owners, end users. We get all those folks together in a room and talk about what species they would want to be able to purchase if they were doing restoration, the species that are of interest to them. That could be everything from, someone from the highway department, and what their needs look like and how that could be very different from, say, a rancher that is focused on livestock production but wants to incorporate natives into their livestock production. And so we get all of those people together and find out kind of the species that are of interest to them. Once we've identified those species, as Shelley said, we go out and make hand collections. It's an individual or a couple of us here in the picture, and we are out picking seed by hand from wild populations. We use a lot of lunch bag brown paper bags. And if you think about almost all of the material that you can buy commercially, and definitely the material that was developed by either Texas Native Seeds or one of the plant material centers, started with a lunch bag size collection of seed. And as you'll hear through the presentation, we go back to that seed and that lunch bag a couple of times throughout the process. The map that's up here on the screen, every one of those dots represents a collection that was made by someone within the Texas Native Seeds program. Once we've identified those species of interest, we try and make at least 2 collections of that species from each county that species occurs in. For some of species, these evaluations can get into the several hundreds of populations. We've just completed a little blue stem evaluation that was up that way. We did a Canada wild rye that was the same way. Conversely, if there's a species of interest to a particular set of landowners that has a much more restricted distribution, that evaluation might be a lot smaller. One of the ones that we're working with right now is a species of southern witchgrass. Primarily grows on the sand sheet in South Texas. That evaluation only contained 12 different populations because it has a much more restricted area of adaptation. As was kind of talked about, we then plant these out in common garden studies. Everything that we do, we start from transplants. So we grow a lot of transplants. We have greenhouse facilities spread throughout the state. Each set of plants shown in this picture represents a different population. As you can see right here in the front, this is a knotroot bristlegrass evaluation. That accession in the center, or that population in the center of that picture, has produced seed much sooner than most of the other populations that you can see kind of in the background or either right next to that. The color's a little bit different. There's more seed there than some of the other populations that are producing seed. The one thing to note too, though, is there's probably not as much leaf material in that population as some of the other ones you can see around. So while all of these are, knotroot bristlegrass, each population's a little bit different. And so as Shelley had kind of alluded to, we really want to be able to pick those kind of top tier populations that are going to be our best seed producers, our best forage producers, have our best seed fill, and be able to work for a lot of different uses. Another example here, little blue stem. This was an evaluation in Knox City. Much more drastic differences between each population. That one on the far left being a bright green color, much leafier. Those 2 populations in the middle, a real kind of bright blue color, already bolting. I'm not sure on the date on this, but one thing that we've seen is that if you start moving plants too far from where they were collected, their timing gets thrown all out of whack. And so they may seed 2 or 3 months earlier than most traditional species would. And you can see up at the top of that picture, right up next to the irrigation system, plants die. We put a lot of effort into keeping things alive. We start everything from transplants, and even doing the best we can, you move some of these things too far and they will still die even as a transplant. Once we've selected the top tiers of populations, we move into a seed increase phase. This makes up the kind of the bulk majority of our day-to-day work. We are taking native species and farming them using traditional agricultural techniques. We go back to that original population, so we're back to that lunchbox and we're starting everything by transplants. Best case scenario, we can start with between 1/10 and 1/4 acre worth of a production field. Worst case scenario, we're starting with 6 or 8 plants. We're fertilizing these, we're irrigating them, we're using herbicides to be able to control weeds, and we have a team of absolutely fantastic student workers that help us throughout the state, coming from multiple universities, that help us keep these things weed free. We need to keep them as weed free as possible so that we can provide the commercial growers with the best possible product for them to get started with. We use multiple different kind of harvest techniques. This shown here in the picture is a flail vac. There's a soft bristled brush in there that sweeps the seed off of the plant. This allows us to make multiple harvests within the same kind of seed set so that we can capture the full extent of that genetic material without damaging the plant, and allows us to kind of maximize production in being able to harvest that seed that is at that kind of perfect ripeness. We also utilize kind of a traditional combine and several other techniques to really maximize seed production. The next phase and things, and that really makes us different, is we work with commercial growers. On a very regular basis, and talking to these guys, we're doing site visits with them. We communicate what we envision is market needs. As people call us and are talking about projects, we're discussing issues we may have had in production, kind of you name it. But the end goal is to help commercial growers to be able to grow large scale production of these things. You know, we're talking in the scale of 600-700 acres of production of one individual release per species. We want to be the organization that is providing the seed material and helping the growers make material available for the pipeline that cuts from the Permian Basin all the way to Houston, or the organization that helps, you know, TxDOT that manages thousands of miles or tens of thousands of miles of right of way to be able to use native seed across the entire state. And so we're really working at finding and utilizing the landscape level impacts of things. And that's, you know, one of the handful of things that makes TNS different. You know, the first one being that we're a private research institute inside a public university. We've been operating for 22 years. You know, we have long-term dedicated funding to what we're doing. You know, as Shelley had kind of indicated, this is not a fast process. You know, from start to finish, we're 7 to 8 years per species. And then we need to maintain that seed stock as well. The other thing being, as part of the university does, is it allows us access to be able to collaborate with a whole suite of different organizations. As I said, we're looking at these landscape level effects. We're not growing a little bit of seed to use it on a restoration project in the first year. We're looking to be able to provide commercial growers with large scale production. And then, as I said, we spend time with those commercial guys and really try and do our best to help them along so that, you know, the public as a whole can be able to do restoration. Thank you very much.
So we are ready to get our last presentation started, and we're happy to have Plant Party's own Tim Sigmund for this presentation. Tim is the Private Lands Program Leader for Texas Parks and Wildlife Department. He was born and raised in Giddings, Texas, and completed a bachelor's degree and graduate research at Stephen F. Austin State University in Nacogdoches, followed by employment with Texas Parks and Wildlife Department in 2009. In College Station, Tim was responsible for 7 counties performing wildlife surveys, public outreach, technical guidance, prescribed fire assistance, public hunting opportunity, wildlife tax valuation planning, and dealing with other wildlife issues. Starting November 2017, Tim began his role as private lands program leader for Texas Parks and Wildlife, dealing with private lands issues in a statewide capacity. Tim resides in College Station with his wife and 3 kids. Thanks for sharing with us today, Tim.
Thanks, Charles. So today I just want to talk to you a little about, we've talked a lot about the seeds, how they are collected, how they're developed, how they're released, how to hand collect, how to do it commercially. And now we're going to talk a little bit about how to actually get them in the ground and grow them. So when we talk about planting wildflowers, what we're really looking at is, everybody would love to have this great big field full of diverse wildflowers. This is a native prairie hayfield in Washington County. But how do we get it there? And so there's some considerations in when you're doing these plantings of how to establish a system you want. And really what you're looking at is not only are you looking at a diversity of plants, but also seasonality. So we have our early season bloomers. This is that same hayfield in May and April timeframe where you have bluebonnets and some of this purple Indian paintbrush and some sundrops and lots of other good stuff that likes that cooler, wetter part of the year. But then we transition into the early summer and midsummer plants, things like Plains coreopsis, annual sunflower, black-eyed Susan, lemon bee balm. And then moving on into the fall, where you're looking at some of these mealy blue sages and thistles and other things that'll bloom in that later summer time period as we get warmer and hotter. And so really there's kind of 5 rules that I follow to establish successful plantings. And this goes for native grasses, native wildflowers, really anything. First thing you want to do is establish your goals. So is your goal aesthetics? Is your goal providing pollinating resources for bees and butterflies and hummingbirds? Is it producing food and a forage base for songbirds and game birds or mammals? You really want to know what you're planning for, and that'll help you determine what your seeding rates will be, what your seed base will be, which plants you want to put out there. The other thing, as we've talked about throughout the day-to-day, is procure good quality and clean seeds. You don't want to be throwing down invasive species that are going to cause you issues or non-target plants. You want to make sure you have good germination of what you do throw out there so your calculations are correct. And this can be done, as Hagen mentioned, with hand collecting or from the efforts of all those like Tony and Dawn and Shelley in buying seeds from a commercial vendor. But procure that good quality, clean seed before you ever get started. And then the third one is once you know what you want to do and you have the seed mix that you want, you need to eliminate or minimize the competition that's there where you're going to establish these plantings. Now, this could be on a field scale level where you're doing a prairie restoration, or it could be an urban garden or a wildscape, or even a potted plant on an apartment balcony. But you want to make sure you have a good soil medium for it to be planted in. Sometimes on a field level, you may have to use chemical applications of herbicides to eliminate non-native vegetation to allow what you want to plant to grow. Other times on a smaller scale, you can use solarization or other methodologies that are available, such as tillage, to open up that bare soil and allow these plants to have a good place to grow. Planting at proper rates, season, and depth we'll go into a little bit more here in a little while, but really you want to make sure that what you're planting, you're not buying more seed than you need to save on expense. You're planting it at the right time, and you're also sowing it at the correct depth, so you're not burying it to where it never has a chance to come up. And then lastly, after you plant, you're looking at controlling weedy competition if necessary, particularly things like crabgrass, some of your annual ragweeds, Russian thistle, and other crop weeds that can be a problem. Typically, it's going to be non-native plants that are going to cause you issues. So when we look at these plantings, we'll be looking not only at the fact that of what you're selecting and then is it site appropriate, but also the stature of what you're planting. So some plants grow tall, some short, some are fairly broad, take up a large area, some are fairly spindly, and you can mix these things to maximize the usability of the space that you're planting them in. And so here, for example, we've got standing cypress and American basket flower. And so standing cypress is more of a tubular flower, which may be more attractive to hummingbirds, as to where American basket flower has more of a composite flower, which may be more attractive to things such as bees and butterflies. Here you can see this bee covered in pollen. Pollen sacs are full. There's on its legs. Other plants like bee balm, it's in the name. It's more favorable to bee species for pollination. You're not going to see a ton of butterflies using this. They will use it, but it's primarily used by bees. And so they'll shove their heads up in there and get to the nectar. But you can also notice other species will use it too. So you can see there's a green lynx spider at the top of that photo in what's using this. So you're going to have many things using these plants that you put out there. Again, another flower, Indian blanket, easy one to do, and butterflies will use these as well. The other thing to consider when you're planting these things is to not only consider the adult life stages of these pollinators that you're targeting, but also the larval stages. So here's a swallowtail butterfly utilizing a member of the carrot family in the wildscape in my front yard. And also a really popular one for a lot of folks to consider is milkweed. So you have the benefit of the flowers and the pollinators they provide access to with their flowers alone, but also the host plant for queen and monarch butterflies. So when we talk about these different plant species and what's out there, not only do we want a variety of plants, but sometimes even a variety of floral colors and characteristics can be beneficial. So some research has shown that hummingbirds are typically going to target red, orange, yellow, or pink flowered plants. Bees tend to have a tendency to favor blue, violet, purple, yellow, or even those that give off certain indicators of UV light. Some plants have basically runways lit up with UV indicators leading to where the nectar is located, trying to attract those species once they're close in. Butterflies, pink, purple, yellow, red, and white tend to be more favorable. And then a lot of folks don't think about moths, but they can be very important pollinators, typically on white and yellow flowers. Other considerations for certain plant species for minor pollinators can also be bats. If you're dealing with some of our desert plants, bats can be an important pollinator species. Beetles, if you've ever seen a prickly pear flowering, there's almost always beetles buried in them, and also thistles. They'll be in there eating that nectar, and they can serve as minor pollinators for those. Wasps will oftentimes visit flowers and can be minor pollinators, as well as certain fly species. So when we talk about seeding and selection, do we hand collect or we purchase? That's really up to you. If you're doing a large seeding, as we do in some of our prairie restorations, typically we're going to purchase because it's very difficult to have the time or the money to be able to pay to be able to hand collect enough seed to seed 100 or 200 acres at one time. If you're doing a small urban garden or you want to include some species that aren't commercially available, hand collecting is a very viable option. It helps to provide that diversity that you may not be able to purchase. It also provides that local ecotype and local things that are adapted to the local soils and rainfall in that region. The other thing you want to do is understand the seeding rate. So if you look at the picture on the left-hand side of this photo, you'll see pictures of Indian blanket and black-eyed Susans. In publications released by the USDA NRCS Plant Materials Center, Indian blankets have about 200 to 250,000 seeds per pound. Black-eyed Susans have anywhere from 1.5 million to 1.7 million seeds per pound. So if you plant a pound of each on an acre, you're going to have a heck of a lot more black-eyed Susan seeds out there than you would Indian blankets. I typically plant in my prairie restorations about 2/100ths of a pound, and I have plenty of plants show up. So a little bit of seed for some of those annual plant species go a long way. So understanding those seeding rates and how much you need can be important. The other thing is to understand seeding depth. If you look at the photo on the right, this is a prairie restoration mix that I plant in Parks and Wildlife Pastures for Upland Birds programs. We have seeds from that large one at the top, which is Eastern Gamma Grass, all the way down to those black-eyed Susans kind of down there in the bottom center. Most of these can be anywhere from surface sown to 1/4 inch deep. The main thing we're looking for is good integrated seed to soil contact. This can be done with broadcasting seed or by drilling it with a no-till seed drill. The main thing is that it's in contact with the soil and that it's not interfered with by other vegetative materials such as leaf litter, dead grass from killing the sod, etc. You want to make sure it's in contact with the soil if at all possible. So for most of y'all, if you've already planted yours, this is a picture from my front yard of some bluebonnet clusters that I planted back in October, and some that naturally came up, and they're already 4 to 8 inches across. So the seasonality of when you plant can be important. A lot of our wildflower species will come up in the fall and then overwinter as basal rosettes, and then rise up once the soil starts to warm in kind of early February to late February, depending on what part of Texas you're in, and then bloom in March, April, and May. Here's another example of some of the other ones that are even later in the summer bloomers, but are already up. So there on the left, there's a whole bunch of little rosettes and a little wildscape there in my front yard here in College Station. In the blue is kind of circled black-eyed Susans, and that red square, that's an Indian blanket that's coming up there. So even though these weren't planted, they're already up and ready to go for when we do warm up here in the spring and early summer. So the planting time for wildflowers is important. This timing is usually more important for annuals. When we seed perennials, a lot of times many of those species won't bloom the first year. They're going to spend the first year growing, maybe flowering a few blooms, but they're primarily going to be focused on establishment and blooming in years 2, 3, 4, 5 down the road. You think of things like rattlesnake master, any of your various gay feather or liatris species, some of your prairie clovers, those sorts of things. It may be a couple years if they're coming up from seed before they flower regularly. But for annual species, if you're trying to plant March through May bloomers, you're thinking Indian paintbrush, bluebonnet, some of those types of things, you're probably going to want to primarily plant those anywhere from late September all the way to mid-December, depending on where you're at. Those will provide those big dinner plate size bluebonnet blooms, are plants that you're talking about that will produce 10 to 20 blooms per plant. If you plant them later, I've planted them all the way into January, but you're going to end up with a small spindly plant with maybe only 1 to 3 blooms on it. May to July bloomers, some of those early summer blooming species like black-eyed Susans, bee balm, plains coreopsis, annual sunflower. I've planted those as late as mid to late February here around College Station and still had them come up and be able to bloom. If I start planting them in March, it has to be a particularly cold year for them to do well. And for those plants that from June through frost bloomers, if I think of things like cowpen daisy, I kind of plant them whenever. As long as they have favorable weather conditions, they're probably going to come up and bloom later in the season if you get some of those fall rains. But usually they too also like to be established in that late September or dormant season. But you can go all the way into early April and still probably be able to get some blooms. Things like annual sunflower, cowpen daisy, Indian blankets are things that are pretty forgiving on when you plant them. So it's not too late if you want to do it after today's presentations to still throw some of the things out there and see results this year. So we're not going to talk too much about individual species because where I live in College Station doesn't look much like Fort Davis State Park here. The wildflowers will be quite different. The landscape's different. This is about 4,800 feet higher in elevation than where I live. So where you're at is going to determine a lot of what species you select. But the main thing is just to get started. When I started this wildscape here in this left-hand picture, those were all planted in the first week of January, a little later than that time period that I told you, but it's just, I had just bought the home and I wanted to have it in there in an area that was fairly dead. On the area on the right, I got it in even later and it was a shadier spot. And so I chose to go ahead and supplement it with perennial potted plants. But the most important thing was I had good seeding depth. And so if you look at that furrow on the left, that's a picture of a drilled area that we did for a prairie reseeding. About a third of that seed is on the soil surface. About a third of that seed is just even with the soil level. And about a third of that seed is a 1/4 inch below. And to me, that is the perfect seeding depth that I've seen have the best level of success with what I've done on about 10,000 acres of prairie restorations over the last decade or so. It's done really well for the multiple species that we plant, up to 40 species at a time, and all different varying seed sizes. We've had good success. The other thing is bare ground isn't necessarily a bad thing. Here's an example of a solitary bee species that has excavated a nest there on the left-hand side of the screen where that little hole is, and you see all that excavated dirt where it dug in after we had that bare soil. So providing that diversity of flowering resources and heights and structures. You can see some giant coneflower there in the back mixed in with bee balm and black-eyed Susans. Early in the summer, this is kind of late May to early June. But then as we go on, this was middle of July. Now we have Texas bluebells and Illinois bundleflower there providing a different a different thing that's there, but you can look right behind it where all those flowers were earlier and they're gone, but we're still providing nectar and pollen for what's available for the plants that are still there. And now into September, and the cowpen daisy is there. And so any bee or butterfly or whatever species, insect species that relies on these plants has something to access during the entire growing season. It's not just over after we mow off the bluebonnets in late April. So plantings take time. We recommend supplement seedings with potted perennials if you have less than ideal conditions. There's going to be a mess. There's a reason they're called wildflowers. Embrace the mess, embrace the wildscape. So plan for those different life histories, annuals and perennials together, tall and short stature, spring and fall blooms, different flower structures, composite flowers, tubular flowers, mass plantings. This is what filled in on where I planted those potted plants with Greg's mist flower. And now instead of the butterflies competing with one another, sometimes I'll have 15 to 30 butterflies at any given time in that little area, utilizing it, including migrating monarchs. So while we all would love to have just a huge 200-acre field of wildflowers and grasses to manage, every little bit helps. And you can stack a lot of diversity into a small area that anybody can enjoy. And so remember those 5 rules for an effective planting: establish your goals, procure good quality and clean seed, eliminate and minimize the competition before you plant at the proper rates, season, and depth, and control the weedy competition if necessary after you plant. Lastly, here's a good list of resources wherever you are in the state to be able to access some good information. So the 9 Natives program by the Coastal Prairie Conservancy is for the urban gardener who doesn't know what to plant. It lists 9 just pick 9 wildflowers and plant those. The Xerxes Society for Invertebrate Conservation has a pollinator resource center. This one's focused on the South Central region, which includes Texas, which has some great resources. Oklahoma State Extension has some great wildflower gardening and Oklahoma tips for those of you closer to the Red River and in the Texas Panhandle. Lady Bird Johnson has an excellent Texas top 20 list for those plant species that grow almost anywhere in the state that you can choose from, and most of them are commercially available. The Texas Native Seeds Programs has a whole list of publications on how to establish native plants in popular and scientific articles at that link provided. And then the Native Plant Society of Texas, if you look them up, has a plant list for every region in the state on what to plant, how to plant it, and where you can find some of those plants available. So with that, I'll wrap up. Thank you.
Thanks, Tim. That was excellent. Thanks again to our door prize donors, Bamert Seed, Corteva, InView, formerly Bayer Crop Science, Renewable Resources Extension Act, Texas Brigades, and USDA Natural Resources Conservation Service. We appreciate your support of Plant Party. Thanks again to our speakers and to you for attending our 9th Plant Party webinar. Again, Plant Party is a quarterly advanced plant webinar training series brought to you by USDA Natural Resources Conservation Service, Texas Parks and Wildlife Department, and Texas A&M AgriLife Extension Service. The next Plant Party will be on March 29th, 2023 on everything pollinators. So bring your learning cap for some spring green up fun. We will be sending an email with more information to all of our past registrants. Thanks a lot for attending, and I hope everyone has a very Merry Christmas and Happy New Year.